We've been working on creating a new biodiversity baseline for the estuaries in New South Wales. So the project is a part of the New South Wales Estuary Network Protection Program, which was created in response to the floods of 2022. What we noticed is we just don't understand how bad the influence was on our estuaries, on what lives in the estuaries. So what we're trying to do is create a new baseline to understand what lives in the estuaries, from the smallest microbe to the biggest fish to mammals that live in the estuaries, to know that in the future, if there's other disasters, say floods, bushfires, so we can understand what changed and how we can manage the effects of those impacts. One of the biggest methods we use is something called eDNA metabarcoding. eDNA stands for environmental DNA, and environmental DNA is something that's everywhere in the environment. It relies on traces of DNA that living species shed. So it's everything from humans to fish to trees, algae, everything sheds DNA in the environment. And what we do is we use a method that's very similar to detecting COVID in the wastewater to detect all of these species. In the past year, we've gone to almost 300 sites. They go from the very north of New South Wales down to the south, close to the border of Victoria. Our biggest collaborator is the New South Wales Department of Primary Industries and Regional Development. And so we work very closely with them to do these surveys. We also work very closely with different Indigenous groups across the state. So at the moment we work with 6 different mobs. It's a nice way to co-create knowledge and co-design where we're going. So the way we collect the samples is we take a few buckets of water, we go to the creek or to the estuary, and we just scoop up a couple of liters of water. The next step is that we filter that water through a small filter, and that filter goes off to the lab where they then extract the DNA and then study where the DNA comes from. The powerful thing that I still think is quite amazing, that from just a bucket of water and a tiny little filter, we can detect thousands and thousands of species. At the moment in our data set, we've got almost 10,000 taxa, so that's a lot of species. A lot of those are bacteria, little microbes that we can use to study, for example, nutrient cycling after big floods, but all of them are also bigger animals that are of interest to fishes, like we're detecting molloway, we're detecting bream, we're even detecting sharks. What we're already seeing is quite a strong effect of the recent floods caused by ex-tropical cyclone Alfred. We went to the Northern Rivers area, we resurveyed a lot of the sites that we had, and what we can see is that the species that live in the estuaries, they really shift with these floods. And so one of the plans for next year is to look at whether the rivers recover quickly and which estuaries recover quicker than others. Ultimately, all of that data will be of use so we can protect our environment and we can manage it better in a way that we can use estuaries in a sustainable way into the future.